All right, cool. So today we are going to be learning about photosynthesis. Photosynthesis is the way that plants obtain energy. Well, plants and autotrophs obtain energy by converting solar energy, carbon dioxide, and water into glucose and oxygen, which, if you remember correctly, are the reactants for cellular respiration. Now, photosynthesis is a dehydration synthesis reaction. It follows an anabolic pathway, which means that it is an endergonic reaction, meaning I need to invest energy into photosynthesis for it to happen. Now, let's talk about equations. So, sorry about that. For photosynthesis, do y'all remember the reaction? It is 6CO2 plus 6H2O becomes C6H12O6 plus 6O2. Now, in this reaction, let's talk about oxidation and reduction. We covered oxidation and reduction in the previous chapter with cellular respiration. In this case, things are going to be a little bit different. Remember, we are looking at the hydrogen to determine if something is reduced or oxidized. So if carbon here is now here, and you see that it has gained, it is now connected to a hydrogen, carbon in this case would be reduced. Whereas oxygen, because we're only looking at carbon and oxygen here, because the hydrogen is what's moving, oxygen here is now no longer attached to a hydrogen. So in this case, it is oxidized. Because remember, oxidation is loss, reduction is gain, or oil rig, as we've been saying it. All right, everybody understand all this?
Yes, sir.
All right. So let's go back to this. Now, let's talk about plant structure, because before we can understand the reactions and the processes of photosynthesis, we need to understand how water gets to the places it needs to be, because you need water for this entire reaction. So if you look at on the right, if you can, if you can see my cursor, we have two parts, the root system and the shoot system. If you remember from your freshman bio class, the root system is the roots, the shoot system is everything above the dirt line. And the root system is where water is absorbed. Water, when it is absorbed, uses capillary action and transpiration. Capillary action meaning all of the vein traveling, and transpiration being how, similar to how water sweats, it causes a suction type movement. It allows the root system to absorb water, and it uses the xylem inside of the plant, which are like its veins, to move water up through the plant into the leaves where photosynthesis happens. In the mesophyll, unfortunately, this model doesn't have the end of a xylem, but in here there would be an end to the xylem, and the xylem would actually, this is where the xylem actually ends in the leaves, and it would send water through the mesophyll, aka this entire interior part as it is labeled over here, into each of these individual cells so that it gains, and so that these cells which do photosynthesis can use the water. Now, another thing that we don't see here is how carbon dioxide is gained. Well, at the bottom, you see these stomas, or these openings. This is where carbon dioxide is taken in by the plant so that the plant can do photosynthesis. So now we have both of our reactions, or both of our reactants accounted for. We are missing sunlight though, but we will get into that with light reactions. Now, let's look at the chloroplast. The chloroplast is made up of thylakoids, the grana, and the stroma. The grana are stacks of thylakoids. Each thylakoid is an individual little thing on the stack, or oval shaped thing on the stack. And the stroma, like the mitochondrial matrix, is the inner soup, as I like to call it, of the chloroplast. Everybody clear on all of this? This should all just be background information.
Yes.
Okay, good. So now let's talk photosynthesis. Photosynthesis, like this picture, consists of two parts. The light reaction, where the sunlight is, well, where the sunlight takes place to convert water into O2, and the Calvin cycle, which turns carbon dioxide into sugar. Now, both of these reactions have to occur in order for photosynthesis to really work and produce its two products and to, get its energy working. Now, with cellular respiration, we saw NAD+. In this case, we are now seeing NADP+. There's an extra P on the end. It functions similar to NAD, but it is a completely different thing. Make sense? All right, let's talk about the light reaction. So in order for plants to absorb light, plants only absorb a certain type of light, right? Off the visual spectrum of light, they absorb every color except for green, which is why plants are green, because if it absorbed green, the green light wouldn't be reflected back into our eyes and we wouldn't see plants as green, right? Plants actually absorb red and blue light. And during this process, there's two types of chlorophyll, right? Chlorophyll A and chlorophyll B. Chlorophyll A works in photosystem 2. Here it's labeled PS2, that's what it stands for, photosystem 2. And here, chlorophyll B works in photosystem 1. That's their difference, right? And if you look at this whole thing, what does it look like? Anybody?
The electron transport chain.
Yes, it looks like the electron transport chain, right? It functions in a similar way. It takes electrons and it moves them through this chain to create a gradient to make ATP utilizing ATP synthase. Now, how does it get electrons and, well, how does it move them? To answer that question, we have to look at water. Water down here, it's kind of hard to see, I apologize for that, but water here, when it meets photosystem 2, it actually breaks apart into hydrogen and oxygen, as you can see here. When it breaks apart, it actually releases an electron into this system, right? The electron in photosystem 2 photosystem 2 then absorbs the sunlight, exciting up the electron, making it move, and it'll move through this system and will essentially follow a similar path to the electron transport chain, except at the end, it doesn't meet oxygen, right? What it does is it meets NADP, and NADP will combine with that electron into NADPH. That makes sense? That is the path of the electron. All right, I'm just going to keep going then. Now, when water splits, it splits into hydrogen and oxygen, right? This H plus is going to form a bit of a gradient on this end, right? And so when all of this happens, right, the oxygen doesn't have anywhere to go, because remember, in this version, oxygen isn't the final electron receiver, so oxygen is considered waste. And so oxygen is released. So now we know where oxygen in the products comes from. It comes from the light reaction. When water is split into hydrogen and oxygen, it releases the oxygen because it doesn't need it. Now, the formation of the gradient, right, because there are H pluses here and H pluses here, right? It doesn't show this on this picture, but there should be a higher gradient of H plus ions in here because on the outside you see ADP is synthesized into ATP. So it follows a similar function. They use chemiosmosis with the gradient to go, to go with the gradient through the ATP synthase and combine the ADP into ATP. So that's how plants get ATP through the light reaction. However, this ATP is about to get used. Does everybody understand everything about the light reaction?
Yes.
Yes, okay, good. So now we have one half of this done. Let's talk about the Calvin cycle. Now this is a bit of a complex picture, but there's a way for us to simplify it, right? So this is the very scientific picture that we use, or that we decided to use for this. But I'm going to draw a more simple version while I show y'all the path. So we need to do something called carbon fixation. Oh. In the process of carbon fixation, we start with carbon dioxide. Your good old CO2 molecule, which I'm going to represent as that because it only has one carbon, first meets up with a thing called RuBP. That is capital R, lowercase u, capital B, capital P. And it has 5 carbons. And the first thing it's going to do is they're going to bind together in a process called carbon fixation. And if we look back on our old picture, you'll see that happening here. So now, oops, sorry, my bad, give me 1 second. So now it'll combine into a 6 carbon molecule. That 6 carbon molecule is then going to be broken into 2 3 carbon molecules, similar to how you would break it apart in glycolysis, but this time they're going to become a thing called PGA. And you have 2 of them. Does this part make sense to everybody?
Does that mean it breaks down into 2?
Yeah.
Okay.
What does the PGA stand for?
PGA. So PGA stands for phosphoglyceric acid, right? Phosphoglyceric acid. Oh, I did miss something. There should be a phosphorus on either end of these. That's where the phospho in phosphoglyceric acid comes from. All right, so this is step 1 in the Calvin cycle, our second half. Ready for part 2?
Yes, sir.
All right, so looking at part 2. Sorry, hang on, I gotta flip my computer back over. In part 2 of the Calvin cycle, we are looking at reduction. Reduction. Well, what are we reducing? In the light reaction, we did produce NADPH. So NADPH and ATP, which we made in the previous light reaction, is going to reduce PGA into G3E. So these two things are going to go in. NADPH is going to lose its H, and it's going to turn into NADP plus. And ATP is going to be spit out as, well, what do you normally get when you use ATP? You get ADP. Ta-da! So I can't really draw this out, so I'm just going to show you guys on our PowerPoint, which it's actually seen here. You invest a little bit of ATP into your PGA molecule, which is shown here, right? Your ATP gets turned into ADP, and your NADPH gets turned into NADP, but in the end, you get this, a 3GP. That is all of step 2. Does this make sense? Guys?
It does.
It does? Okay. So along with making a 3GP, it is also going to make part of your glucose, right? It is going to make a, what is it? C2H4O2 molecule, which is only part of C6H3O6, right? Well, 1/3 of it. But it can't actually make any more off of that one carbon. Or sorry, no, sorry, I gave y'all the wrong molecule. It's a CH2O molecule, right? Only 1/6 of a glucose. And it can only do that because, well, it only took in one carbon. But you see, it's a cycle. Well, now I got to remake my RuBP. And that is step 3 of the Calvin cycle. The Calvin cycle needs to, well, regenerate its RuBP. So let's go back to the whiteboard. When we regenerate RuBP, right, the first thing that needs to happen is 3 cycles need to happen. So I may have gone a little too fast there. So we took in one carbon and we produced 1 3GP. What needs to happen next is another carbon needs to go through this cycle and produce another 3GP. And then a third carbon needs to come in and produce another 3GP. And when we have enough 3GP, which means 5, right? So every 3 cycles, we combine 5G3P into RuBP. In this process, we also make 3 of the COH2O, or CH2O molecule. Sorry, I keep saying it wrong. And well, now we have half of a glucose, but we also have all this leftover 3GP. What are we going to do with it? We're going to recombine it to make RuBP so that more of this cycle can happen. So now we go back to the whiteboard. So now we do step 3, the regeneration of RuBP. In this process, you're going to take 6 G3P. G3P looks like a 3-carbon molecule. Right. Every single cycle produces 2 of these, by the way. I forgot to mention that. Every single cycle produces 2 of these 3-carbon molecules. So after 3 cycles, I have 6 of them. And what's going to happen? I'm going to combine 5 of them. I'm going to release 1 as part of the sugar. So this G3P is going to become part of our eventual glucose product. Well, the other 5 are just going to recombine into ρBP. And when they recombine into ρBP, well, you get our old molecule. 1, 2, 3, 4, 5. The original molecule, which combined with the carbon. Does that all make sense?
Wait, so Marcus, the
Yes.
G, sorry, because it kicked me out for some
Oh,
reason.
okay, that's fine.
Yeah, but for the G3P, there's now 6 of that? That's a 6, right?
Yeah, sorry. Every single cycle produces 2. Every 3 cycles, we're allowed to regenerate ρBP because we need 6 3G3P, right? So if we need 6 of them and each cycle produces 2, we need to run the cycle 3 times in order for us to have 6, and then the 6 can become ρBP and spit out a 3GP into our sugar.
Okay, thank you. I'm just going to copy it real quick.
Yep. So actually, every 2 regenerations of ρBP, you're going to make a sugar, right? Because if I do this twice, I have 6. And if these 2 combine, they form a 6 carbon sugar. So every 2 cycles, or every 2 regenerations of ρBP, you should get a sugar. This is why you need 6 carbons in the photosynthesis process. Does that make sense?
Yes, and I'm done copying, by the way. Thank you.
All right, good. So that is the final step in the Calvin cycle. So let's do a quick review. Step 1. Let me go back to our slides here. So step 1, a stray carbon is going to combine with our ρBP and merge together, or merge together into a 6 carbon molecule, then break apart into PGA, or 2 3 carbon molecules called PGA. During the reduction process, ATP is going to be converted into ADP, and NADPH is going to be converted into NADP. We got the ATP and the NADP from the previous light reaction. When they turn into G3P, which is the end product of the reduction part of the cycle, you end up with 2 3 carbon molecules called G3P. Then every 3 cycles, you get enough G3P to spit one of them into your sugar and take the other 5 and recombine them into ρBP. That is, again, every 3 cycles, aka every 3 carbons you invest, gets you 1 ρBP regenerated and 1 G3P, which means you need to do 6 cycles of the Calvin cycle in order to produce 2 ρBP and 2 G3P, and those 2 G3P combine into 1 glucose. Does that make sense? I know it's a lot.
Wait, can you repeat that? You said 6 cycles of the Calvin cycle to produce.
Yeah. Every 6 cycles of the Calvin cycle makes 1 glucose. So now, onto a bigger subject. This, by the way, this entire process is considered C3 photosynthesis. C3 photosynthesis has a problem with it, a very big problem. It's called photorespiration. Let me write out the word for you guys. Photorespiration. Photorespiration happens when your RuBP molecule, instead of binding to a CO2, like a good molecule, it binds itself to an oxygen. And the reason why this happens ties back to earlier, right? Earlier when I talked about plant structure, a rewind time a bit, we saw the stomata on the bottom. The stomata, well, they have the job of absorbing carbon dioxide. But what happens when they close and they're not absorbing carbon dioxide? Well, photorespiration. Photorespiration can occur in this case. And the problem with photorespiration is that it wastes a lot of energy for the plant. So when those stomata close, the RuBP binds with the oxygen and it isn't able to make energy properly. Now there are special types of plants that can prevent this type of photorespiration, because you don't want to waste energy. As an organism, wasting energy means dying. So everybody have this down. I'm going to clear it now. So we have two different types of plants that we can talk about, C4 and CAM. Let's start with C4 plants. Now, when I talk about C4 plants, I don't mean the explosive. I mean plants. C4 plants separate their light-dependent and their Calvin cycle physically, right? Because remember, RuBP only binds with carbon dioxide in the Calvin cycle. Oxygen shouldn't be anywhere near the Calvin cycle because oxygen is mainly a byproduct of the light reaction, which occurs in the thylakoid. The Calvin cycle occurs in the brain fart. Occurs in the stroma, the inner soup, right? So they shouldn't be anywhere near each other. But in C3 plants, oxygen from the light reaction can interrupt the Calvin cycle and get mixed into that whole process if the stomata closed. So if they are separated physically, why did I switch back? I meant to, my bad, guys, my bad. So if they are separated physically, like a C4 plant does, right? What happens is, well, your light reaction can do its thing in a completely different part, and your Calvin cycle can happen in a completely different part. Since they're physically separated, it minimizes the amount of oxygen that is coming into contact with your Calvin cycle, which prevents all that photorespiration happening due to a physical barrier. Some examples of C4 plants can be crabgrass, which I might not know about, and sugarcane, and corn, not maize, corn. Now, the second type of plant is the CAM plant, as you can see here. CAM plants, rather than, or CAM, this stands for Crasulian acid metabolism, as the scientists like to flex with their big words. Let me write this out for you guys. Crasulian acid metabolism. They separate the light-dependent and the Calvin cycle by time. Let's go back to our old model. In this model, we see, well, the Calvin cycle occurs during the day, and the other processes occur during the night, so that it minimizes the contact between the oxygen and the Calvin cycle, right? So in this process, it doesn't really describe it, but the reason why it's called the Crasulian acid metabolism, or CAM plant, right, is because CO2 is absorbed during the night. And as it is being absorbed during the night, it is formed into an acid. That acid is then used in the Calvin cycle, right? So now there's really no reason for oxygen to be in anywhere near contact with the Calvin cycle. And this process is mainly performed by desert plants, your cacti. So that's actually the end of all I had to really cover. Elsie cream. That's really all I had to cover today. So are there any questions on photosynthesis, C3, C4, and CAM? No?
I don't have a question, but can I see the chart thingy again that you
Oh,
just?
yes. For C4, CAM?
Yeah.
Yeah. Okay, there you go. So if that is all, don't forget to hit like and subscribe. I do appreciate the subs. Miss Simonis, if you wouldn't mind dropping a comment on this video, criticizing me. I actually really like the criticism. Thanks you guys for watching. My name is El Sukram. I'll see you all on the new server.